Yeah, so this is our project and story about how calcium homeostatic feedback control predicts atrial fibrillation, and in particular its remodeling and kind of its long-term progression. So first I'll give you a bit of background about the heart. I'm not sure how familiar everyone is with stuff, but if you heard it before, it's fine anyway. So this is a general anatomy of the heart. You have the atria above that take in the blood from the veins, they pump it into the ventricles, and then the ventricles pump it to the rest of the body. And the atria, this is where the normal cardiac impulse originates. Somewhere here you have the sinoatrial node. That's on the back wall, the posterior wall of the right atrium. And this is what we'll be talking about today. So really just the atria, that's what we're interested in. So you have the right and the left atrium, and then this is a beautiful drawing I made. So you have the sinoatrial node somewhere in the right atrium, and the property of the sinoatrial node is that it generates impulses on its own, it oscillates. So you have these electrical impulses that oscillate with a particular frequency that can be modulated by the nervous system. And in the healthy state, what usually happens is you have the normal wave propagation through the atria, through both the right and the left, they're connected. And the two atria are somewhat isolated from the ventricles. You only have passage through a small connection called the atrioventricular node that modulates this conduction from the atria to the ventricle, and then the electrical activation spreads from the atria to the ventricles, and you get ventricular depolarization that pumps the blood. So this is kind of a sketch of how the electrical activity looks in the sinoatrial node. So it's oscillatory, and also you have calcium that also oscillates. Usually in the healthy state in phase with the voltage. So basically the voltage is a signal for the calcium, and the calcium oscillates with the voltage. And the calcium is also very important in the working muscle because it is the signal to contract. So basically that's the end effect of this signaling, the whole signaling thing is that you want the muscle to contract. And the atrial myocardium and the ventricular only conducts impulses. So you have, it receives an impulse, it depolarizes, you have this plateau phase, and then it goes back down to resting state, and it doesn't go off on its own usually. So the atrial muscle conducts the impulse, but doesn't generate it on its own. And again, you have a calcium transient that oscillates with the voltage that tells the muscle to contract. And if this whole thing is working properly, you have an impulse in the sinoatrial node. It flows outwards in the atria, and then the atria contracts so that they pump the blood, and so do the ventricles.
A quick question, just to keep my head on straight, if that's okay.
Yeah, sure.
The calcium waves that you're talking about, I assume that is intracellular calcium. And
yeah, so these are all single cell, like the sketches I made. It's for a single cell, please.
And that is calcium coming in through ion channels as opposed to, calcium becoming released intracellularly?
So they have both. So cardiac cells have kind of like muscle cells, like skeletal muscle cells. So you have a calcium current that is a membrane current. And you also have intracellular calcium stores in the sarcoplasmic reticulum, which is a variant of the endoplasmatic reticulum. So you have.
And so those waves are both releases put together.
Yeah, basically that's the sum of the cytoplasmatic calcium, which comes in from outside and comes in from the inside as well.
Got it. Thank you.
Yeah. And you also have like calcium-induced calcium release. So the if the endoplasmic reticulum senses an elevated calcium, it releases on its own. So you kind of have a feed-forward mechanism. So it releases a bit of calcium, and then the whole thing triggers. Yeah. But compared to the skeletal muscle, it doesn't have nerves. So that's the main difference. It just conducts the impulse on its own. It doesn't have a nerve for every fiber. That's the main difference. Yeah, so in a 2D tissue, and this is what we'll be looking at a lot in this presentation. It looks something like this. So you have normal conduction. You have an impulse generating spot here. In our case, it's going to be artificial. We just induce a current in these cells here. So this is a 2D tissue with a number of cells. And you have a wave propagating outwards. And you have the color scheme here for the voltage. And the main focus of the presentation here is when this goes wrong. So we have arrhythmias. So you'll have something that's called atrial fibrillation, where instead of one single coherent wave that beats every, once per second, so 60 beats per minute, as you have the normal heart rate, you have all these random chaotic waves that are very fast. So the activation is going to be much faster than regular. And you also have, because of this, you'll have disordered contraction. So you'll have the blood will stagnate in the atria and you'll get clots, and those clots can go to the brain and you get strokes. So it's really a major health problem as well, because you have people that get disabled for life, so it's really a very bad disease if you get it. Yeah. And the interesting thing about atrial fibrillation is that it's been known for some time, like from the early 20th century, that it is progressive. So you have this chaotic fast electrical activity that by itself leads to long-term pathological changes in tissue. And these changes are in the form of electrical remodeling. So you have changed expression of ion channels in these cells, and you have structural remodeling. So you can have fibrosis and atria enlarge. So you have mechanical problems. And these changes then make atrial fibrillation more likely to occur. So you have, at first, you have intermittent episodes, and then they get more frequent. And in the end, you have permanent atrial fibrillation, where you never have normal conduction. And what's interesting here, which has been shown in 1996, 95, is that the electrical activity alone can change the substrate. So what they did is a famous experiment is that they took goats. It's usually done with large mammals that have a heart closer to human. And you just pace them. So you implant a pacing electrode and you pace them very fast. And what this does is that the electrical activity alone changes the substrate of the tissue. And then after you pace them for a couple of days, then they just have AF on their own. You don't need to pace them anymore. They're already in the chaotic state. And what's interesting is then this gives you kind of a natural history of AF. So you have this time progression over long time scales that we're not usually used to dealing with. Like these simulations, people usually do them for a couple of seconds, milliseconds, something like that. So the natural history goes from tissue substrate dynamics. So you have protein expression, fibrosis, things like that, proliferation of fibroblasts, whatever. You go to depolarization waves and the action potentials in the cells. So you have, seconds to milliseconds level events, channel gating, and all that kind of stuff. And these changes then feed back all the way back into the substrate dynamics. So they change the underlying substrate. So you have basically this coupled system that works over, months. That's kind of the time scale. And in a kind of historical analogy, if you want to have an analogy for this, you kind of have the geography of a country or whatever. You have some long-term structures appearing, such as cities or settlements, trade networks, so on, that feed into day-to-day life of the people there. And this feeds back into the city. So the way the people are living there, if they're prosperous, they go build their city better, and you have more trade and so on. So that's kind of a historical analogy for all of this. And what we're interested in here is to look at how does this whole coupled system, as a coupled system across these time scales, evolve, and specifically how does atrial fibrillation initiate and then progress. And now to get back to more some more technical stuff, what we're working with here is simulations of cardiac electrophysiology using models that probably you're familiar with. You have the Hodgkin-Huxley model. That's just the classic formulation, just the scheme. And this is specifically the model that we're having here. So it's called the Comanche model. It's specifically of the human atrial cell. And you have all these currents that are known to be in the atrial cell. And to go back to the question earlier, you have this intracellular compartment with calcium. So you have all this calcium cycling represented in the model as well. And what this really, I highlighted it here, is that you have an explicit representation of the intracellular calcium. So you can track it as well over time, not just the voltage. And this is kind of the mathematical formulation that we'll be using here. So you have the derivative of the voltage. You have all the ion channel currents, all of these here with their gating variables, just the standard Hodgkin-Huxley formulation. But we'll also have this diffusion term for 2D tissue. So it's basically a PDE. And you'll have diffusion factor. So basically, because the cells are connected to their neighbors, voltage diffuses outwards and it activates them. And that's how you get the waves. And really, calcium is interesting because it's the link between the beat-to-beat dynamics, so the slow, the fast dynamics of action potentials and the beat-to-beat, seconds, milliseconds time scale, to the long-term regulation. And you have all these complex intracellular events. So basically, the idea is that you have fast pacing that drives intracellular calcium overload because you just stimulate it a lot. Calcium gets accumulated in the cell. Just by the fact of stimulating, you have higher average intracellular calcium. And through these kind of complex intracellular regulatory networks, through all kinds of transcription factors, the cell really tries to maintain its homeostasis. So you'll have electrical remodeling, changing channels, upregulating or downregulating expression. And you also have structural remodeling, as I said. But really, the point is that the cell tries to maintain its calcium. So it's going to lower its calcium currents. It's going to increase calcium channels taking calcium out of the cell, such as NCX. It's going to shorten the action potential because the length of the action potential gives you the length of the calcium transient. So by shortening the action potential, so repolarizing faster, you'll have less calcium entry in the cell. So all of these changes, the cell at the individual level tries to maintain its homeostasis, but at the tissue level, it makes arrhythmia more likely. And we'll see how in the next couple of slides. So you have this complex intracellular network, but really and it's the same as in the neuron, for example. And this paper, so by O'Leary and colleagues from 2014, proposed a very simple feedback model that kind of summarizes, it's kind of the set point of the whole network. You just assume that you have a single calcium target, that's your feedback target, and then M representing mRNA and gamma representing scaling factor for the channel conductances will change accordingly. So if you have intracellular calcium above this calcium target, you're going to have a reduction in channels, or, depends on the channel. Some of them go up, some of them go down. But the idea is you have this feedback model that tracks a single calcium target with just these two equations, kind of putting all of this network into a single, into just a couple of equations. And this is the O'Leary paper. So that's where they first, feedback models for neurons were, they've worked on them since the 2000s or even earlier, but this is kind of a very simple, elegant way of looking at it. And they described you can have different types of neurons, that you have emergent activity in them, and you have different, pacing patterns in these neurons. They also talked about network homeostasis, where you have multiple neurons and you perturb them, but you have this feedback system that adapts to whatever perturbation you put in the network. So this was the original paper where we took the feedback model. And a couple years ago, we published this paper. I'll have a couple more slides at the end if we have time to talk about it. Where we show the same thing for the sinoatrial node. So we adapt this model for the sinoatrial node, so for the oscillatory activity that I showed you at the beginning. And we see that we have naturally emergent activity. So from single cells, if you make the conductances very low, they're quiescent, and then they pick up and start beating on their own. And in the tissue, you have self-organization into different centers where the impulse originates. And these are all things that you can see both in the development of the heart and in the adult anatomy and function of the sinoatrial node. So really the idea is that you have this feedback model that's kind of a model of homeostasis. So usually its purpose is to maintain homeostasis and keep things in a physiological level. But in our case, in the disease case, it's pushed into a pathological state. So just to reiterate, we coupled this feedback model to the human atrial cell, that model in particular that I showed you. And it's going to span multiple timescales, so from milliseconds to hours. And what we do here, and it's novel, is that we simulate up to 24 hours of continuous 2D electrical activity. So we have this tissue where we look at millisecond events, but we simulate it up to 24 hours of simulation time. And really CUDA and GPU really made this possible, because otherwise you couldn't have done the time scale here. So this is basically our electrophysiological model. As I said, you'll have variation in voltage at any particular point in tissue. You'll have the diffusion that makes it a 2D tissue. So every cell is connected to their neighbors, and it's going to be coupled to the feedback model. And the math looks something like this. So you have the intracellular calcium target is the set point of the regulatory network. Intracellular calcium is that value that oscillates with the beats. And that's going to translate into an intermediary that's equivalent to mRNA expression. And that's going to go again to this gamma that scales the maximal conductance for each channel species X. So that's going to control the maximum amount of current flowing across the membrane. It's basically equivalent to how many of those channels you have on the membrane. And then you have these taus that are some time constants that control the sensitivity of each of these to its particular, the right-hand side of the equation. And the interesting thing is that if you have different ratios in this tau x, you're going to control the ratio between the different gammas, because gamma just reacts to m. So however you control m, you're going to end up affecting the gamma concentration. And in our case, x is going to be all of these channels. I just put them here. It's more interesting if you're actually a cardiac physiologist, but I just have them here for reference. So this is kind of the first results of the model. You're just going to have a single cell at first. I want to see how it responds to pacing. So the baseline of the model is that all of these gammas, so all of the scaling factors for the different currents, start at 1. And we chose the calcium target such that at 1,000 millisecond pacing, that's equivalent to 60 beats per minute, you have all of them stay at baseline. So that's basically your healthy state, your regular heartbeat, human heartbeat, that's beating and maintaining the tissue within normal parameters. And this looks something like this here. So we have the voltage. This is the actual simulated voltage from the Corbinci model. So you have the action potential. And then you have the calcium as well. And here on the right. And 1,000 milliseconds is the cycle length, so distance between 2 beats. So as you increase the pacing rate, so you make this shorter and shorter, so you activate them faster and faster, you have more and more remodeling. And this really happens because by pacing them faster, you increase the calcium in the cell, and then the cell responds by remodeling. So you have changes in all of these channels, and these succeed in taking the calcium, so the average calcium, this is kind of the thick black line here, they go back to the target. So it succeeds in maintaining its homeostasis, even up to 250 milliseconds. So that's like 200-300 beats per minute pacing here. So as you pace them faster, you get more and more remodeling, and the whole thing is reversible. So if you go back to 1,000 milliseconds, back to 60 beats per minute, they all go back to their baseline of 1. And this is just the calcium target into that line here, so you can see the average always coming back to it. But these changes lead to changes in the electrical phenotype of the cell. So the action potential duration and the resting membrane potential are things that we look at in cardiac cells and that are known to change in atrial fibrillation. So the action potential duration is this, how long the plateau is basically. And because of the changes, you're going to have a shorter action potential. And you also have a somewhat lower resting membrane potential. So that's where the baseline of the electrical activity. And further, this also leads to reduced calcium transient. So this is, again, the target here. And you can see that the remodel, so you have the comparison between the baseline model and the very remodeled state that's paced very fast. Oh yeah, and just before I go on, so here, just to be clear, this is over hours, right? So this is, we simulate these beats that happen every second or faster than that, every 250 milliseconds, but we simulate the whole thing over hours. So you can see kind of the time response. It takes about an hour for the ion channel remodeling to take place. So during this whole 5-hour simulation and even longer, as we'll see later, we always simulate all of this activity. So the beat to beat, second millisecond activity. So that's basically baked in here. If you zoomed in here, you're going to see a lot of these oscillations. Right. So going back to the remodeling, if you have these changes at very fast remodeling, you're going to have a change in the electrical activity and also in the calcium. So the calcium really succeeds in maintaining the calcium target. So you can see that it's centered on this target that the feedback model imposes. And here we introduce another concept of co-regulation. So all of these channels, they don't change alone, basically, is the idea is that if you have a change in one, you're going to have a change in others to compensate. So you don't want to change just one channel or another. You want to kind of change the whole of them in a kind of coordinated fashion so you can get functional electrophysiology once you change them. And what this gives you is, first of all, a way to derive the ratio of these changes from experimental data. So you have basically the known expression of the channels in the healthy state, and you have the known expression of the channels in atrial fibrillation. So we know that all of these channels change in channel expression, in patch clamp experiments, and so on. So we know, and due to these two factors, you get basically a slope that gives you the value in atrial fibrillation and the value in the healthy state. And we assume that these changes will happen linearly on a slope. So either from the healthy state to the atrial fibrillation state. And this is basically the relationship between all of the channels. And what further this allows us for the presentation is that we can represent everything by the one of the channels. So for the value of one of the channel gives you a unique set of all the others, basically. So you can represent all of the other channels by just one. And some of them go up, some of them go down, but you always have this unique relationship. So we basically impose this ratio on the model. And the first interesting result of the model is that you get at fast pacing overall levels that are similar to those known in AF. So we don't impose the actual level that they end up being remodeled at. We only impose the way they vary together, right? So we pace them very fast, as happens in atrial fibrillation or in experiments. And we get to a level that's somewhat close to the actual level seen. So like half, for the example, for the calcium current, it's about half the one in the healthy state. So we get that. We see it here. It's close to 0.5, the green blank here. So that's the first result of the model. Just by imposing this constraint, we get accurate or qualitatively accurate expression changes. And finally, this is just one calcium target, right? We picked one that stays at baseline when you pace it at baseline pacing rates. But you can move, obviously change this. So if you have a lower calcium target, you're gonna have more remodeling because the cell has kind of a lower inherent set point. So whatever you stimulate, it's gonna act kind of extra because of it. It's gonna be more driven to remodel. And on the other hand, if you have a higher inherent calcium target, you're gonna have less remodeling. So the cell is gonna be kind of more resistant in a sense to pacing and calcium overload. But the linear relationship, so the thing governing how these channels express together, is always the same. So you always have this kind of the lines here, but you fall on different places depending on your calcium target and on your pacing level. Yeah, so that's kind of the single cell stuff. And now the more fun stuff is the thing in the simulations in the tissue. So what we see is that, first of all, you have this, we're going to start with the homogeneous tissue, where we're going to have the same calcium target at all spatial locations. So you have basically a 6 by 6 centimeter tissue represented here, and that's 512 by 512 cells. So each pixel basically in this tissue represents a cell, and each cell has an individual calcium target. And in this case, they're all homogeneous. And what we see here is that we pace and we try to induce a spiral wave. So this is kind of a very common thing we do for cardiac simulations. And at first, the tissue is resistant. So we can induce a spiral wave, but it's going to be unstable. And this is, again, something that's known to happen. So in healthy tissue is resistant to reentrant activity, as we call these spiral waves. And if you look at it here, not this one, this one. So you pace in the corner, and then you have this induction of a spiral wave, but it meanders a lot, and it hits a wall, and it dies off. And you try it a couple more times, it's always unstable, and then you have pacing resuming. And the important part is that you pace between these tries. So you pace the tissue very fast. And this drives remodeling, as in the single cell, but in this case at the tissue level.
Can I throw out a quick question again?
Yeah, sure.
So trying to remember my understanding of AF, and what I'm remembering is that, and it sounds like this may not, well, what I'm remembering may not completely be correct, but what stops a healthy heart from having these spiraling waves was that tissue always conducts bidirectionally, and with bidirectionally conducting tissue, you can never have a spiral because, you know, eventually you'll have two waves going different directions and they'll collide at a piece of tissue that's still refractive, and the spiral will die.
So you can have like self-standing spirals, but with the thing with it here, so if you have perfectly homogeneous tissue, is that you have to convince it to do a spiral wave. So that's why you do, so if you see here, you can have this cross stimulation. So you have a wave going from top to bottom, and then you have this square here that kind of induces the spiral wave. But if the tissue is remodeled enough, you can have self-sustaining spiral waves. You can have like a traveling wave, I mean, except periodic conditions and stuff like that. But
all right. So that's the point then, is that because of all the remodeling, the tissue is no longer homogeneous.
so no, the tissue is enough if you have enough remodeling. So basically, the action potential duration is the key. Because if you think about it, if it's shorter, then the cell can activate faster again after an activation. So it can sustain this wave kind of circling around itself. That's the idea. And the remodeling drives the APD changes. So you don't need any imposed heterogeneity at this point.
Okay, got it. Thanks.
And so you have the pacing between the cross stimulation, and basically we'll have two main time scales of the remodeling. So we'll have the time scale of the ion channels that you see here in green, but we'll also in the tissue, we'll also remodel intracellular coupling. So that's basically represented by that diffusion coefficient. And each cell in the tissue basically has its own value for the diffusion coefficient. And that's also remodeled in the same direction as the channel. So it goes lower as you have more pacing, but it's going to be even an order of magnitude slower. So you basically have electrical activity, you have electrical remodeling that happens on an hour or so, and you have structural intracellular coupling remodeling that happens on 10, 12 hours in our model. And once you get enough re-entry, let me see if I can. Yeah. You get a stable spiral wave. So kind of after 1 hour of pacing, you get the spiral wave that just does this on its own. And the most interesting thing here for us is that because the spiral wave is localized, it's in this spot, it creates a remodeling pattern on the underlying tissue. So again, this tissue is perfectly homogeneous from a parameter standpoint. It has the same calcium target. It doesn't have anything distinguishing one part from the other. But because the spiral wave stays here, the cells at the very center of the spiral wave, so you can see we track the tip of the spiral wave here, the cells actually get activated less because they don't get so stimulated. It's always somewhere in between, right? The spiral always circles that area. They don't get fully deactivated, but they don't get fully activated either. And you kind of get this remodeling pattern emerging from the tissue. So these cells underlying the spiral wave, from their point of view, they're better than the others because they get activated less. So they're going to be healthier in a sense. And all the rest of the cells get paced very fast by the spiral wave and then get remodeled, equivalent kind of to the fastest remodeling we had in a single cell. So that's just the homogeneous tissue. And in the long-term behavior, we also see the behavior of the diffusion coefficient. So that also goes down, but on a lower time scale. And this here marks the first hour of simulation that I showed in the previous slide. So we're going to have the stable spiral that stays stable for a long time. And obviously, we don't pace, we don't intervene anymore once the spiral is stable. And that kind of maintains this remodeling pattern over a long period of time. And looking at it in this video, this panel here tracks the location of the spiral wave, kind of as a color map, as a time average. You have the electrical remodeling, and you have the intracellular remodeling. And it's going to be stable. So this is over hours of activity, right? And at some point, because of the intercellular coupling, it's going to start to drift slowly. So that's kind of the behavior that we're seeing. Yeah, so we have the idea is that we have both ion channel and structural remodeling. And finally, to go to actual atrial fibrillation, we'll have a heterogeneous tissue that's bigger. So it's going to be 12 by 12 centimeters. And it's going to have a heterogeneous calcium target map because these cells are known to be heterogeneous. So they always have difference in calcium transients, always has, if you look at them at the tissue level, they have differences in voltage. So there's some kind of inherent heterogeneity in the system, right? And in this case, in the atrial, in the heterogeneous tissue case, which is equivalent to kind of 1,024 by 1,024 cells. We're going to have just pacing. So we're going to pace in the corner. And we're going to see that this causes reentrant activity on its own. So first of all, looking at the long-term behavior in this heterogeneous tissue, we're going to have a phase where the AF gets initiated. So the orange lines mark pacing episodes. So you see at first we pace it a lot and remodeling progresses. And then the pacing episodes become rarer and rarer. So as the algorithm, basically the pacing works, if it senses kind of continuous activity and reentrant waves, it stops pacing. It lets it be and evolve on its own. And this leads to the duration of the AF episodes becoming longer, kind of on a log scale here. So at first they're slow, they stop and start. And at some point here, it goes on for the rest of the simulation. It doesn't stop anymore. And this is just an example of pacing and of reentrant activity. So you kind of get a feel for what the activity is like during those times. So the first part of it is initiation. So how does this initiate? And this is the very first reentrant episode in this tissue. And if you look at the video here, you have pacing in a corner, you have voltage here and calcium. And this is the electrical remodeling. And this is dynamic, but on the time scale of this video, that's like 20 seconds or so, you won't see any change in it because it changes too slow to see it in a video, right? So you have this pacing in the corner, and at some point, you have this kind of spiral wave breakup here around an area of heterogeneity in the tissue. And because of this, you're going to have kind of spontaneous reentry. Now the pacing stops, and you have kind of continuous reentry. You have these spiral waves moving around and generating more spiral waves. So you have this chaotic activity. And because the tissue is kind of not remodeled yet or enough, it stops on its own. So it kind of hits a wall, they extinguish, and then we restart the pacing. Here, we restart the pacing. Again, you get reentry, and so on. So you do that for the whole 24 hours of the simulation. So this leads to AF progression. So over the 24 hours, you're going to have, by counting how many spirals you have in the tissue, how many reentrant waves, we're going to see that you have more and more of them over time. And it's kind of a biphasic response. So you have a sharp increase corresponding kind of to the electrical remodeling. And then you have a slow overall increase corresponding to the structural remodeling. Over time, it also increases the dominant frequency shown here. So you have these, basically how fast the activation is going to increase somewhat over a long time too, and how fractionated the activity is. And it also organizes. So the spiral waves, if you look at where they're located in the tissue, so just by tracking where each reentrant wave is, over time, you kind of have, especially in late, very remodeled tissue, you have this organization where they find a spot and they pin to it. And in that spot, you're going to have this kind of tissue niche, in a sense, where because they stay there all the time, they remodel the tissue. And again, you see the same behavior as in the homogeneous case, where the core of the spiral is less remodeled. So it's kind of healthier because the spiral always stays there. And looking at it in a video, so here on the left, we'll have where the spirals are. You're going to have electrical remodeling and structural remodeling here on the right. So you can see at first they're kind of diffuse and disorganized. And as the simulation progresses, they get more concentrated. And at some point here, you're going to see that they form kind of these stable spots here. And then they're a bit, not forever, but then again at the end, you're going to have another stable spot here. So that's where there's always a spiral. That's the idea. And looking at this more closely, so this is just 20 seconds of activity of very remodeled tissue at the end. If you look at all the spiral traces just on their white coordinate, basically, you're going to see this stable reentry kind of highlighted in orange here. And that's somewhere here. You see the kind of the bump that it creates in the tissue. And you're gonna have, if you look at the local activation map, you're gonna see that this spiral, the one that's stable, paces the area around it very fast. You have a very high spot right at the core of the spiral, and this whole niche that it creates. So again, the tissue, the substrate reacts to all of these changes. So we have these, the bump is completely emergent because of the spiral is there. It's not caused by the heterogeneity. And looking at the cells, you have the voltage and the calcium in red. The calcium is in red. And you see kind of this chaotic pattern caused by the waves. And this is a video of this activity. So this spiral, the one that we're talking about, is like here, you always see it's always there. And the other ones kind of come and go, but that one is always there. And it kind of creates its own environment. And finally, if you have different heterogeneous maps, you're going to have different types of behavior. So some of them you have intermittent activity where you never get full-time atrial fibrillation. In some of them you get permanent re-entry, that like the one we saw, and some of them are completely resistant to re-entry. And this kind of explains some of the behavior in human patients too. Like some of them have just intermittent episodes of atrial fibrillation, some of them get permanent, some of them don't get it at all. So in conclusion, we have integrated this long-term regulatory model based on calcium feedback with the human atrial cell. And we really join multiple directions of research. So we have tachypacing leading to fibrillation, remodeling, intracellular regulation, ion channel co-expression, so they behave in a coordinated fashion into a kind of a single theoretical framework. And we have these very interesting emergent behaviors. So we have spontaneous re-entry initiation, and especially the formation of these tissue niches patterned by the spiral, by the activity itself. So you basically capture all the fundamental aspects of the natural history of AF. And kind of for future work and why I wanted to reach out in the first place, you can imagine easily like substrates that sustain kind of these emergent kind of objects, if you want, such as these spiral waves. And you're going to have long-term patterns in these tissues, not just cardiac, so in development and so on. But the interesting thing that we add with this is that you have activity and substrate co-evolving. So you don't just have some pattern that forms. You have that pattern that changes the substrate, and then the substrate changes the pattern, and so on. So you kind of have the system co-evolving over long time scales. And ultimately, presumably, this leads to homeostasis in the tissue structure, but also, as we saw here, leads to pathological cases. And it's a very general thing that, you know, pretty much every biological system spans multiple time scales that interact across it. So we can have wide application. And again, this is kind of summarizes everything. And with that, thank you. And these are our papers so the Sinai journal paper that I mentioned, and this one is a preprint, and we just submitted the review, the revision. So hoping it's going to be out soon too. Yeah, thank you.